Hi everyone, it's Mr. Sinti, and today I'd like to talk to you about the process of ligation. As you may know, that what we've been doing so far in the lab is that we've extracted DNA from the redwood, we've pulled out the genomic DNA, and what we've attempted to do is to amplify just a portion of all of those genes using a technique called PCR, which is really capable of mass producing, making many copies of a particular region because we were using primers to do so. And so we used our friend the thermal cycler to do that. And so the next part of the workflow of the lab is to then take all those PCR products and then insert them into something called a plasmid, which is going to act as a cloning vector so that the DNA can be amplified or propagated even further. And so though that may sound confusing, that's the point of the video, to discuss that. And so what I didn't mention was the fact that once we amplified the DNA, we also did something called PCR product purification. If you recall, we ran the PCR products through a column using size exclusion that where these little tiny beads were able to capture the unincorporated DNTPs and maybe some primers as well, and polymerase. But hopefully the PCR products, all those pieces came out, and so we have amplified DNA. And so what we want to do is take the DNA and basically do this. This picture right here is, let me come back here. Well, let me go forward. A typical bacteria is something that we're going to be using, and we're going to be using the bacteria E. coli. And why would we do that? Because we want to make many copies of our redwood DNA. And one of the things about it is, a little background on E. coli is that it has this big circular, one big circular chromosome. And it has many, although there's only one shown, many small circular rubber band size loops of DNA called plasmids. And here's an actual picture of that circular plasmid under the transmission electron microscope. And so what we're going to do is use these plasmids, and then here's another picture of the plasmid, and this one's a little bit more coiled up. And so basically what we're going to be doing is taking that rubber band and putting inside of it our redwood DNA. And so we're going to use the plasmid as a vector. And so it's called a cloning vector. This part right here is the cloning vector, this part right in here. We're going to use this plasmid as a cloning vector. And what we're going to try to do with the plasmid, once we've inserted the redwood DNA into it, is we're going to then try to put the plasmid into an E. coli. So E. coli. We're going to put it into E. coli. And if this plasmid gets incorporated, this bacteria will be transformed because it never had that redwood DNA in it before. So it's transformed. And the neat thing about it is that the E. coli is very good at dividing rapidly. And so in so doing, it'll make many copies of our DNA. And so we'll then be able to purify the plasmid from those bacteria and then sequence our redwood DNA. At least we hope to. And so as I was saying, the plasmid is a much smaller piece of DNA. It's usually circular, and it has just a few genes on it right over here, and we're going to add our redwood DNA. Now, of course, redwood DNA never normally exists in a plasmid. So we're going to be recombining redwood DNA that we pulled out, and this is now going to be called recombinant DNA. So we pulled the DNA out, and we're going to recombine it with a totally different species. Now, of course, not only is it a different species, an entirely different kingdom, and I would even go as far as say it's a different domain, because this is a eukaryote and this is a prokaryote. But we're going to combine the DNA from a redwood into bacteria so that we can clone it. Okay, so that's what we're going to try to do. And the point of putting the redwood DNA into the bacteria so that the bacteria can mass produce and we can grow it in the incubator, and it grows really quickly, and it'll make many millions of copies, not only of itself, but of our redwood DNA. And so some of the background about what we're going to be doing. And so plasmids what do they normally do? Well, they normally carry genes that make bacteria resistant to antibiotics. And so what we could do is use that. In other words, we could select a plasmid vector. We can select one that, for example, has ampicillin resistance to it. And so let me just draw that just in case it's confusing. So we can choose a plasmid vector that has a gene already on it for ampicillin resistance. And so if we could put that into the bacteria and grow it on petri dishes that have ampicillin on it, we'll know that those bacteria are the ones that we're interested in, where those are the ones that are transformed, and so those are the ones that have redwood DNA, because normally the E. coli wouldn't be resistant to ampicillin. So we can call that a selectable marker to indicate whether or not we were successful. And so let me go ahead and do that. So if you had the plasmid like this, and if you inserted the redwood DNA into it like this, and over here was a gene right here for ampicillin. Okay, so this is our plasmid. And it's like, well, part of what this conversation is really all about is how did we even put the redwood DNA into the plasmid, and that's called the ligation. That's what I want to talk about, but this has to be discussed first. So if we were to successfully ligate the gene into the plasmid, we would know so by transforming it. In other words, like putting it into a bacteria. Okay, so we have to transform the bacteria. In other words, make sure that it has our little plasmid in it. Okay, here's ampicillin. Now, how do we know that we're successful at doing so? Well, we would have to grow the bacteria on a petri dish that had agar on it. Okay, so this is growing medium with ampicillin. And if it had ampicillin and if it was able to grow, then we know that we would be successful. That's one way that we could do it. And so another way that you could screen, although we're not doing this in this particular one, but I thought I'd point it out, another way that you can screen your bacteria to see whether or not they've been transformed or not is sometimes they have what are called reporter genes incorporated in the vector plasmid. In other words, they have the lactose operon, or they have a gene that produces a green fluorescent protein called GFP. And if they have the lactose operon in it, what you could do is induce them to produce beta-galactosidase, which is the enzyme that breaks down lactose. And so if they do so, you can add a chemical called xgal, and then the colonies will turn blue as a result of it. And so long story short is that you could also determine whether or not you're successful growing your bacteria if you used the chemical xgal in your growth medium. The colonies would actually grow blue. And likewise, if you were using the green fluorescent protein gene, your colonies would produce green fluorescent proteins, and you would know, hey, these are the ones that are transformed, as opposed to others that might be growing that are not. And so this is how you can tell that this is your desired product right there. So just saying. So some of the newer plasmids that are useful in the lab have what are called a positive selection mechanism. And so check this out. So here's a plasmid. So it's a circular piece, and this happens to be the one that we're using. It's called P-jet. So P for plasmid, P-jet 1.2. And it is at exactly 2,974 base pairs in length. Okay? And so what's interesting, you see this purple area right here? And notice how the plasmid has been cut open. So it's a rubber band cut open. What's interesting about it is what we would like to do, as I mentioned before, is we would like to take all of these PCR products, in other words, all these little pieces of redwood DNA, and put them into right there. We would like to put them into the plasmid and use the plasmid as a vector, and then put that into a bacteria. Now here's the deal, that if you do not put your your insert into this vector, what will happen is the plasmid will just close upon itself. And what will happen is that purple gene right here, which is sort of like half of it's over here and half of it's over here, is called ECO471R. Okay, and you're like, what is that? So what's interesting about that is that gene produces an enzyme that actually kills the bacteria. It produces a restriction enzyme that goes and cuts up DNA, and so it'll actually destroy the bacteria. So if the bacteria do not grow on petri dishes, if there's no bacteria, then we know that they don't have redwood DNA in them. And if the bacteria, so this is kind of simple. So if the bacteria grow, we know that the gene is actually in there because we've disrupted this lethal gene. And so the lethal gene will cause cells that were not transformed to die. And so I hope you're able to follow that. So how about this? So if the bacteria are growing like this, if you see individual colonies like this, you're like, hey, those have redwood DNA in them. Because we know that we're successful. If DNA is successfully inserted, then the lethal gene will not be expressed. And so this is called positive selection. And so, what up with that? Well, it saves us from using reporter genes, like, in other words, the beta-galactosidase and green fluorescent protein, although those are kind of cool too. And so what's fascinating about this is that plasmid, if I go back to the plasmid just for a second, for the plasmid to be able to produce ampicillin resistance, and for it to really get going, in other words, don't want to get too technical, but for RNA polymerase to really start transcribing the foreign plasmid, what's important is that you grow the bacteria on a petri dish, because we are incorporating the lactose operon into the plasmid. We're not going to use it as a reporter with the excal, but what we're going to do is when you grow the bacteria on a petri dish, not just with lactose, but with a chemical called IPTG. This chemical will cause the plasmid to replicate more readily. Okay, it'll do that because it causes, and let me explain how that works briefly. This is somewhat complicated, and so the chemical IPTG, let me write it out, IPTG. This chemical is going to be growing and is going to be added to the petri dish. And so what this does is it acts as an inducer. And so this is a gene on our plasmid of interest, okay? And what that chemical is going to do, it's going to bind to a repressor protein and make it inactive. The repressor protein is something that prevents the plasmid from expressing itself. And so we want to make the plasmid express itself. And so what we're going to be doing is adding that chemical, which is going to then bond to the repressor protein, derailing it so that RNA polymerase can go its happy way and start transcribing all of the genes necessary in the plasmid. Okay, and so let's talk about now the process of ligation. Now, ligation is just simply inserting the redwood DNA into the plasmid vector. And so, it's one thing to insert it, like, okay, you put it in, but it's like, when you're transferring something like in a document, and you're going to cut something, which is what we're basically doing, is cutting a piece of redwood DNA and then pasting it. If you're going to paste it, you're going to need paste, or you're going to need tape or something to get the piece to stick into the plasmid. And so what's fascinating is there's an enzyme called DNA ligase that actually helps to incorporate the redwood DNA into the plasmid. And so the DNA ligase, the enzyme that does that, is something called T4, and that's named after a bacteriophage. If you recall this, A bacteriophage is something that is a virus that infects only bacteria, and it's named T4, so that's where that comes from. And so basically what's happening here is that here's our plasmid right here. And so we want to, again, take our redwood DNA and place it into the plasmid. And so what we're going to be needing is this enzyme right here, DNA ligase. And so DNA ligase is going to come along like this and then paste it in. It's going to come along over here and paste it in. So that's DNA ligase, the enzyme that actually incorporates the new piece with the other piece. So there's some detail to that I'm leaving out. But I will say this, that the enzyme requires ATP, and it requires a little bit of magnesium ion, Mg+2, for it to work properly. So this is going to be in the buffer along with the DNA ligase. So when you're pipetting that, you'll know that there's three things in here: the enzyme and these two ingredients. And so another thing of consequence is that the concentration of the vector, which is the plasmid, and the insert has to be proper ratios for the ligation to be efficient. And so as it turns out, it's not very efficient. And so let me talk a little bit about the two ways in which you can get a piece of DNA into a plasmid. And so let's say this is our redwood DNA. One way of doing this is by cutting it, because again, if you're going to put something in, you have to cut it. And so traditionally the way it's done is that a piece of DNA is cut with a restriction enzyme. That's an enzyme that cuts DNA at specific spots called restriction sites. And those enzymes were taken from bacteria. And so if a redwood DNA was cut with this, it would leave, do you see how if it was cut here and here, it would leave a single-stranded DNA as what's called a sticky end. And so this is our DNA fragment right here, okay? And this is of the plasmid. So let's do this. So here is the plasmid, like that. Okay, and so remember I said the plasmid is a circle, and I said, well, if you're going to put something in it, you have to cut it. And so this is what we mean by cutting it open. And so it's going to actually have on the end of it this little sticky ends like this on the bottom and on the top right there. And so this right here is our fragment. This is our redwood. Okay, this is our redwood DNA. And it normally needs to be treated with the same restriction enzyme so that it can incorporate itself into the plasmid. And then we would add, well, how does it do that? Well, it just naturally hydrogen bonds, but in order to get them to covalently bond permanently, that's why you need the enzyme DNA ligase to do that. And so this is called sticky end ligation. All that being said, we're not going to be using restriction enzymes to insert our redwood DNA because it requires enzymes to do so, and so we could do it a little bit easier. We can use something called blunt ends. And so blunt end, really briefly, I'll describe it more, is that the plasmid that we're working with is already cut. And so it's no longer circular. One can say that it's actually linear since it's already cut. And so it's blunt, meaning like it doesn't have any ends to it, it has no sticky ends. And so basically the redwood DNA, I'm switching colors now, the redwood DNA is just going to incorporate directly into it, although you still need the enzyme ligase to suture it in. And so here's a little illustration showing like these scissors are like restriction enzymes. If you recall, we used restriction enzymes when we were discussing DNA fingerprinting. And you can cut DNA with restriction enzymes, and then you'll know that they're cut if you run them through an agarose gel, and you can see the restriction, fragment length, polymorphisms. So here's a cut with a restriction enzyme that manifests into sticky ends, and then this is a cut that results in blunt ends, right here, a straight cut. This is the kind of ligation we're going to be working with. So our plasmid has a blunt end, okay? So here's the thing, however. So here's our plasmid, and we want to put our redwood, which is shown here in red, we want to put our redwood into this. And so you would think all we need to do is just simply put a plasmid, pipette our DNA in, and add the enzyme buffer, and it'll do its own thing, and it will. However, there's one little step that has to be done before the ligation. As it turns out, that the PCR that we were running with Taq polymerase, this is a very useful polymerase because it's heat sensitive, so it's really great and it's readily available, but it has a little bit of a problem. It's not a proofreading polymerase, which means that it doesn't, it makes kind of a lot of mistakes, but it also doesn't go back and check itself. And as it turns out, on the 3' end of our little fragment, let's just say right here, it actually leaves a little overhang of the nitrogen base adenine. And so that one nitrogen base would prevent the ligation of our redwood DNA into the plasmid. And so what we have to do is simply like cut that little A off of it. And so that's what we call blunting. So we want to make our fragment blunted so that it can enter, so it can enter into the plasmid in the blunt end of the plasmid vector. And so how are we going to remove that little A right there? What we have to do first is that we need to treat it with a proofreading. So we're going to add a DNA polymerase before we do the ligation. So the DNA polymerase is going to come and it's going to snip off that adenine. It's going to proofread that and turn it into a blunt end. And so once the A has been removed, look, there it is being removed. So now that's called blunting. So now we're going to be able to then ligate the fragment into our vector. And so why use this particular vector? A couple of reasons that this particular PJet 1.2 vector is useful is that it has blunting involved in it, and so there's no need to be using an excessive amount of restriction enzymes. Another useful thing that may not be understood until later is that once you insert the redwood, let me go here, once you insert the redwood in here, do you see this little green area? This little green area has many regions of DNA, and all these little letters and numbers are different restriction enzyme sites. And so what's fascinating is that it's called multiple cloning sites for restriction enzymes. And so what we're going to be doing is once we pull our plasmid out of our bacteria and purify it, one of the ways that we want to check it to make sure that it's working is that if our redwood is in here, we should be able to cut it out and run it through a gel. And if we cut it out and run it through a gel, we can cut it here and cut it here, and it would produce a small piece, and the rest of the plasmid would be a big piece. And if we have two bands on our gel, we'll know that we're successful in our entire workflow. And so that's pretty useful, though kind of complicated. It also, a good reason, I mentioned this before, a good reason to use this plasmid is that it has ampicillin. Another good reason is that it has this lethal purple gene right here. So if the plasmid were to stick to itself, so in other words, there's no redwood DNA being put in there, what would happen is it produces an enzyme that will actually kill the bacteria. And so any bacteria that are not transformed simply die. Another good reason to use this particular vector is that it's commonly used, and we know its exact sequence. And you're like, why is that important? That's very important because that's what we're trying to do in this whole workflow is sequence this piece of redwood DNA. And if we know the sequence in front of it, like over here, and we know the sequence behind it over here, we'll then know where the redwood begins and ends. How about that? Although it's harder, it's actually harder to do than it sounds. So here's one of the things about the ligation now. The ligation is kind of inefficient because I'm making it sound kind of simple. You get your plasmid, and all you have to do is put in your redwood DNA and add the enzyme ligase, and it's like, there we go. But as it turns out, the success of it is only like 1 in 100. And so that's not so good. And so we're just going to be hoping for the best on that. And so chances are on our side, though, because there's a lot of bacteria that we're going to be working with. There's going to be a lot of product. That's the reason why we did PCR, is to have lots of fragments of redwood. And so we're working against it, but we're also with it. So there's other possible products other than the desired one. And so how about this? What would you say, and I mentioned this before, what would you say if there's something called self-ligation? In other words, the plasmid decided to just simply connect back to itself. And if it did that, do you remember? What would happen is that would mean that these two pieces would come back together, and that would make a complete gene, and that would be lethal. So that this won't happen, because the bacteria will die if that happens. So another thing that can happen that's not so good is if we ligate with some primer dimer. And so primer dimer, remember, is the primers that we worked with in the PCR. And you're like, well, didn't we remove those when we did the PCR purification? We removed most of them, and that's why we did. Because one of the things about primer dimer is that this attaches to DNA like this, and this is a poor drawing, but it attaches forward and reverse. And one of the things about it is that's what you want it to do, but sometimes it actually anneals to itself and it makes double-stranded DNA, and then this starts amplifying in the PCR reaction. And so this thing could get incorporated into the plasmid as well, and that's not good. Or what could happen is more things can happen is the ligation can actually have multiple inserts. And so check this out. So here's our plasmid, and we're like, yeah, we got our redwood DNA in there, and then we also got our redwood DNA in there, and we also got our redwood DNA in there. And so this is not so good too, because overall this isn't going to be the desired product. Like, we don't want many copies of it in there, and it's going to reduce the ratio of our insert to our vector, and it's ultimately going to screw up our possibilities. Because in other words, if you get like 3 into this one, the chances of what you really want is many of them that are successful, like this, because this one that goes into a bacteria will actually make many copies, and so this one you're just using a lot of our product, and it's kind of wasting it. Okay, and then self-ligation of insert. Now this is kind of crazy too. Like, for example, if you had, here's your redwood DNA product, and then here's your redwood DNA product, and it's like, well, how's that going to happen? Well, if we're adding the enzyme ligase, it could do that. It could just simply start sticking redwood DNA together instead of it being incorporated into the plasmid. That's not good. And so here are the possibilities, and pictorially maybe this is a little bit better. And so just as in conclusion, the plasmid could self-ligate, but that means that the bacteria ultimately, when we transform them, will die, because this is a lethal gene. We could have multiple fragments. That's what I was just talking about, multiple inserts. This is not so good. We can even have the redwoods ligating to itself. That's not good, because then we don't even have the plasmid, and so that's not going to be able to transform the bacteria. And then over here we have PCR fragments, like primer dimer. That's not good either. So down below here is the desired effect, that we could insert our one copy of our redwood into every single PJet plasmid. That'd be awesome. And so I hope you're able to follow this, and I appreciate you watching it. And I guess the fun of it is actually doing it. So you can look forward to that. So I hope you enjoyed this, and thanks for watching.